the introduction. Yeah, so I was kind of talking with Maria, and we thought like, well, she said it could be kind of nice to give a presentation about size spectrum models, which is what I've been working in the past years, but also like review a bit what are the recent developments and what is the direction that some of these models are going, and some limitations that I have encountered while developing these models or working with these models and data needs. So that's what I will be talking about today. And oh, I cannot switch. Let's see. No. And so I was first going to start by making a quick review of some of these models. And then I was going to switch to a project that I did during my PhD, and then talk about data limitations based on this project. But I realized that it was a bit too much of a detour, and I wanted to promote more of a discussion, so I have removed this part. I hope you're okay with that, and center on all the other topics. I just want to say that first, in this talk, I was like kind of struggling, like what would people expect about when I say size spectrum models? So here, I want to say that I'm just gonna center on these numerically solved mechanistic models with several discrete sizes. So I will try to not look at the NPZ models, but some that just have a lot of sizes that could potentially generate size spectrums. And here I will also center on the ones that are implemented at the global scale, which are often the ones we try to compare with data and maybe use to make predictions or different scenarios or understand processes at the global scale. So when I was thinking on plankton size spectrum models at the global scale, I came up with this. I thought I could divide them in 3 groups right now. The first group is where I would say most plankton models fall. And in general, what they have in common is that they have several nutrients. So we could maybe call them biogeochemical models, global biogeochemical models. They simulate phytoplankton dynamics and zooplankton dynamics, actually. And they also have like the ocean circulation implemented in general, and they have seasonality. In these models, the zooplankton are simulated just like phytoplankton, so it's like some kind of box models with many boxes, and some of them also include functional types. So this is a case of the MIT GCM that is often known as a Darwin model, where they have, I don't know, pico phytoplankton, like prokaryotes, coccolithophores, diatoms, etc. But not all of them have them. At the other extreme of these models, we have sorry, can you see my mouse, Maria? Can you see? Yeah, okay. We have these more zooplankton-centered models, and especially as far as I know, it's one of this group by Ryan Hennigan and company. And in this case, they do center a lot in zooplankton. They simulate zooplankton ontogeny, which basically means simply the life cycle of zooplankton. And that is because zooplankton grow in size during their life. And they can change up to 2 orders of magnitude in terms of carbon mass. So that has several implications for predator-prey mass ratios, predator-prey interactions, growth rates, physiological rates. And we think it can be quite relevant to take this into account for mesozooplankton. And at least what they did was center on the functional types, and but they don't have, well, actually I will go more in detail in each one of these models. And then afterwards we have this in-between model that is the one we developed that does take into account ocean circulation, phytoplankton dynamics, but in this case we only have one nutrient, which is nitrogen. But we do simulate zooplankton ontogeny, and I will explain again in more details. So as I said, just going through these groups, I think I've mentioned most of this stuff, and I think these are the most common global biogeochemical models that can simulate size spectra. And then, as I was saying, this model, even though well, it has very nice things. So the implementation of several zooplankton functional types, as you can see here, we have like larvaceans, omnivorous copepods, carnivorous copepods. Oh, so okay.
Camila, I'm sorry.
Yeah, I know. So, someone, wait, now I can mute. I thought I could mute someone. Well, I will continue. And the interesting thing of this model is that by implementing zooplankton functional types, you can get actually quite different dynamics in terms of food web. So, for example, gelatinous zooplankton have very big predator-prey mass ratios compared to other zooplankton, such as copepods. And by implementing this, you can get some food web shortcuts. So food webs that would, in our models where we only simulate one kind of zooplankton, be very long. Certainly here, they find like some food web shortcuts where actually by having this big predator-prey mass ratio, you can just like directly channel the energy from phytoplankton or small phytoplankton to large zooplankton and perhaps fish. I think the group has some preprints on that. I didn't put it here actually. But these models also have some limitations. For example, they don't simulate phytoplankton dynamics. So in this case, they use some kind of satellite-based chlorophyll input, where they also take into account some size classes. So that's what they take into account as food for fish. And then they also use temperature to affect the rates of the zooplankton. As I said, the ocean circulation is not resolved. So they simulate in each cell with the chlorophyll and temperature inputs. And often there is no seasonality. It's taken at the as an annual mean. And this, I think, also applies for most of the size-spectra models of fish, which I'm not going to center here. As I say, zooplankton is simulated, but they don't, at least in this specific model, they don't really close a life cycle. I think they have like some kind of seed starting concentration, and then the zooplankton just grow. And I think this method is also applied in other models. And they also have the fish dynamics implemented. So yeah, then if anyone has questions or wants to say something, feel free to just interrupt. Okay, I mean, I will open discussion afterwards, but yeah, just feel free to do so. And then finally, we have this a bit, as I said, the intermediate model, which is the one that we developed. We call it the NUM model, and it stands for nutrient unicellular multicellular. And we simulate several phytoplankton trophic strategies. So we have many sizes, and they can be either complete autotrophs, mixotrophs, or heterotrophs, depending on the environmental conditions. We only have one nutrient pool, which, well, we can say that maybe for some regions where iron limitation kicks in might not be the best. Ocean circulation is resolved, but we don't really use a fully resolved the physics of an ocean circulation model, but rather we use what's called a transport matrix, which it simulates a transport between bins in the model, let's say, but yeah, the physics are not explicitly resolved, and it's nice to use this because it's just faster to run. Yeah, we have zooplankton ontogeny, and we do have the life cycle closed, so they have to fully reproduce. And then we differentiate with feeding strategies, active versus passive. This is relevant because one is like more sit and wait and doesn't spend energy, while the other is actively looking for food and spends more energy. And we don't have fish, but an interesting thing that I didn't think about it when I was developing the model, but actually realized is kind of relevant, and you will see why afterwards, is that we resolve the size spectrum of the detrital particles, and especially for the ones of zooplankton, where the fecal pellets do contribute to carbon export and the biological carbon pump, and they can sink quite fast depending on the size of the producer. And then we also have what we call here dead falls, but it's simply dead phytoplankton or zooplankton. And this, we also consider it size resolved, and that all of this can affect the sinking rates. So this was a bit the introduction for the what we currently have of like the latest kind of models at the global scale about size spectra. And then, yeah, I went into all this part of what some people call model validation, which maybe it's not the best word, because I mean, we don't always need to have like the model fit the data to investigate a process, let's call it this way. So I don't think, well, I will open this discussion afterwards about model validation. But I personally kind of struggle with this, and because, I mean, we have a global model, and suddenly, because there is much legacy of the biogeochemistry, we really feel the need to validate. But in ecology, it's a bit harder, I think, to get data because it's hard to measure. And especially when it comes to compare the model with data, I think I was personally a bit naive when I did this project about 2 years ago or something like that. That I wanted to compare like point by point, like I'm showing here. And I mean, it's good. It's a very transparent way of comparing the models, but when you think about it, I mean, we are maybe not really comparing point by point, because to begin with, we have a very coarse resolution. I have, I think, 2.8 degrees. That's like several hundred kilometers, I think. So I'm using like some kind of mean of that area compared to a specific point. Data is scarce. And also, I mean, here, for example, I'm using some kind of climatology to simulate the model. So which is like some kind of mean across seasons, and this is like the model is seasonal, I mean, but I mean, I don't take into account, for example, different years or interannual variability in terms of El Nino or La Nina. So if different samples are taken in one of these different years, even though it's in the same location, they are going to look very different. So I will leave this question as a discussion point later, like what could be the best way to compare models versus data? And I will go now into the data limitations, because this was one of the reasons that I also struggled. So to begin with, as a modeler, I thought, oh yeah, phytoplankton is basic, like everyone models phytoplankton. There must be plenty of phytoplankton data and phytoplankton carbon. But that's actually not the case. There are very few studies that really try to measure from the field phytoplankton carbon biomass. And that is simply because it's very hard to separate phytoplankton from zooplankton, for the microzooplankton. And I mean, it's very time consuming, it costs money, and well, and if you add mixotrophy there in the pool, then you can get wherever. So, okay, so we don't really have phytoplankton carbon data, but we use chlorophyll, which is, I think, what most people then use. So I guess in this sense, we should, even though most of our models have been now going towards simulating carbon to understand better what is, well, the carbon cycle, maybe we should center still on trying to capture fairly well chlorophyll in our simulations, because this is what we have most data, at least as a proxy for phytoplankton. Yeah, microzooplankton data is also a bit of a tough one. So perhaps the kind of data I was getting, and I will talk a bit afterwards, was, for example, this that I used in my paper, where they simply got like all the nano and picoplankton data from the cruise. So that includes like all phytoplankton and microzooplankton up to a certain size. But I mean, of course, here I'm just limiting to a few transects, and actually there is kind of a big, which I didn't know the explanation, well, I put it in the discussion, but like this transect, the MT12, the data, which are the dots, are kind of higher compared to the other two. And I don't know why. I don't know if it's a methods issue, if it was really like this. So we do need like some kind of unified way of sampling if we want to validate models. And then the other data set that is available in this case for mesozooplankton is this copepod data set. And I mean, I think it's okay. I think the data, it's very nice that someone tried to put together like all data available in the world that you can find in one place. But it's also hard because, I mean, it's, as I say, all kind of data sampled in all kind of ways with all kind of methods. Given in very different units, you have like carbon mass, abundance, volume of water displacement or something like this, you know. So, I mean, you have all kind of units. And then, I mean, these people try to kind of put it all together and correct for all these things and put it all in a common, in common units. But I mean, I think the uncertainties on this data set, at least this pooled kind of data set, must be very, very big. And this is something that needs to be addressed. And I mean, I think the data set is still useful if you are picky, if you really go into it and you say, okay, I just want the data that is provided in carbon mass or in whatever, in number of individuals, and is measured with this kind of net on this depth range. So yeah, yeah, so I'm struggling a bit with using this just entire data set as a way to validate our models. Yeah. Then something that I feel like modelers haven't got, well, or at least I haven't seen it much being used in models, and I'm actually getting quite a big fan of, is this particulate organic carbon data. And it's a bit different from what we in the modeling community, at least, or I was thinking in the modeling community. When I thought about POC, it was detrital particles. But these people, when they go into the field and they collect POC, is everything. So they collect everything that is there, phytoplankton, zooplankton, and the detrital particles up to a certain size. I think, I don't remember now how many microns. If anyone knows up to how many microns it is, let me know. But actually there is a significant amount of POC data taken using the same method. So that's kind of nice. And actually, so this is some like data compiled in 2017. I could imagine that now there is more data. And actually satellite methods do pretty fine at least in the open ocean to obtain this, well, POC, I think. I mean, you can argue with that. But so I think that in terms, if we are able to identify these pools of carbon where satellites do perform quite well, we can really use them to compare, at least up to a certain degree, with our models. So I think we should pay more attention to the satellite community, especially because they are going, like they're putting a lot of effort now on conveying uncertainties, which we should also learn about that. And especially next year, they are going to launch the PACE mission, which, well, in short, they will be able to get hyperspectral data. And this will allow to improve the estimates of many of these carbon pools. And this will also, actually, I don't have a slide about this, but it will also, there is a group working on obtaining size spectrum from satellites. And I don't know exactly how well is the performance about it, but they are really working a lot with field work and data people to get the good size spectra. And I think in a few years, they could be getting quite reasonable results. And regarding the size spectra, I mean, we also have size spectrum data. But the problem is that many studies can take size spectrum parameters or can measure size spectrum at different ranges. And this is something I was just showing in our paper, for example. This is, where is my mouse here? So this is in the same location, just different times of the year. And I think it was in North Atlantic. So we first have something that fits quite well, the size spectrum. The dashed line is a fit. And the gray line is like the total size spectrum is just, I just shifted it up to so that it doesn't overlay the other things. And then we have here a bloom, and suddenly we have this like curvy size spectrum, at least in this range. The bloom persists, and then it's again more towards summer or end of summer. We again have a fit on the power law. And then I was hearing many times like people going to the field and saying, yeah, but we measured the size spectrum and it doesn't fit the power law. I mean, like if you fit to, for example, only a phytoplankton, which is this yellow line, yeah, and I try to fit here something, I will get like a straight line probably, something like this. And this is also, for example, what is shown in this study by Niels Jens, where they went and measured with flow cytometry and flow cytobots in 4 different locations. No, I think, yeah, 4 different locations, and the colors are different times of the year. And this is more oligotrophic regions, and here you go into towards more productive regions. And you can see again, this exactly the same, this kind of bloom. And this is in terms of diameter. So it's more or less the same size range that we have here in terms of mass. And they say, yeah, sometimes we do find a very nice like power law fit. But yeah, if you go here and you measure this within this size range, you're not going to get it. And this is something that they already said in the study of Atkinson et al., where you need to measure a kind of wide range, like in this case, mass range, to really get reasonable parameter values, in this case of the slope of the size spectrum. So if you measure a small size range of like with particle sizes, you can get almost anything, whereas the larger the size range you sample, the more you will converge to some kind of theoretical value. And I have to say I didn't know about this data set. I found it while looking at for this presentation, but there is this new data that came out last year, which is actually a size distribution data set of particles using this UVP5 camera. And so they have pulled all this data for different depth ranges. And it's again the same method, so that's kind of cool. And I think it can be a good way to use to validate our models if we resolve them by size classes. The thing is that in this case, at least my experience is that we always use carbon mass, whereas when you go to the field, you measure more length. So maybe we should also try to center a bit on this, whether we implement this part of length in our model so that we can compare it better with what is observed in the field. But again, here, all particles are included, also the trital particles. I don't know exactly what is the contribution of the trital particles to total plankton biomass within the size range in the surface ocean, but it should be taken into account. And so yeah, it's a very nice data set, but if we want to use it in our models, we might need to resolve a bit better the trital particle size distribution. Yeah, so for data limitations, that's a bit what I have. So very few data of phytoplankton and microzooplankton because it's hard to get in the field. Maybe to overcome this, we could use this POC data, and there are it's like a good alternative, even though it pulls everything, but we can technically just use that in our models by pulling everything as well. I think satellite measurements are fairly, field measurements are are quite good, and satellite algorithms also perform fairly well. So I think they are a good way to use in our models. And then there is this UVP5 data set for particle size distribution that just came out that we might be interested to take into account. In terms of model limitation, I mean, there are many. There are a lot of model limitations, and we could go into all directions. And I mean, in the end, we use the models for what we want. So depending on what is our question, we'll prioritize one way or the other, of course. But it seems that we are going more and more towards like this global simulation style of more similar to the biogeochemistry field, that we really want to use our model to make case scenarios or predictions. So we kind of need to, to get like more reliable patterns, or at least patterns that allow us to compare with data. So I just centered with two of them, and we can discuss afterwards on what do you think is needed. But one of them is what I said is models do not incorporate the detrital dynamics. Actually, the detritus changes in size along while they sink or whatever. They can aggregate, they can be fragmented, they can recycle, and this changes the size, and this affects the sinking rates and the residence time in the surface ocean. And as I said, it might be relevant to simulate with field data, and also to simulate carbon export, which is, I think, one of the things that most modelers are, at least ecology modelers, are working on, or at least they put it into the context of the biopump. So yeah, maybe this is something that we should start looking more. Some people have already started working on it. But I think this is something that is generally lacking in these more ecological models. The other question, and it's the way I separated the size-spectra models that we have today, is the ontogeny story. And the question is, do we need to account for the ontogeny of mesozooplankton in our models? And then there is this group of people that kind of already tried to address this question, even though they did it in a 0D model, so it was not really a global simulation. And what they found in their study was that the size formulation generates, so by implementing the size formulation, we could get some different dynamics than if we didn't. Some of them are a delay between the moment you have a pulse of food and the moment you have this increase in zooplankton biomass, because in the moment you have a lot of food, they will gain a lot of energy, reproduce, but they will need to grow again in size. So you will have these delayed cycles between phytoplankton and zooplankton pulses. The interesting thing that they found as well, which I found it kind of weird, but not weird, I didn't know what to think about, was that even though you get different dynamics of zooplankton, these ones didn't really affect the phytoplankton or the nutrient pool. So maybe if you are centering into like a more biogeochemical model and phytoplankton, based on these results, you would say, okay, I don't need to complicate myself and put zooplankton ontogeny, But maybe if you are trying to get whatever fish biomass or link it to trophic transfer, maybe you might want to consider having ontogeny. But I mean, this was, I think, just one kind of test they did. But as I said, maybe when you put it into a connected model where you can have feedbacks with the sinking nutrients or carbon exported or higher trophic levels, maybe this might change whether we have ontogeny. So it's still a bit unclear whether we need that. So some conclusions here. I will give some extra final remarks, and then I will open more discussion points. So yeah, we currently size-spectrum models that we have at the global scale either start or stop at zooplankton. And I think some groups are already looking into how to connect these two kind of models to get from phytoplankton dynamics to fish. Some zooplankton some models have zooplankton ontogeny. It's still unclear to me how relevant it is to do this, but probably just case dependent on what you're looking for. More reliable data sets are emerging and can help validate our models. But yeah, models also need to adapt to what we can sample in the field. I mean, that's clear. We cannot expect just the fieldwork people to sample what we want. And so we should adapt our models to be compared to what is sampled. And also, I want to, as I said, we should really start looking a bit, paying attention to the remote sensing. And I think we can learn quite a lot from them because they are really looking for ways on how to convey uncertainties in a unified way. And I think if this is a path we are going towards comparing with data, we should also be able to do that. I will leave these questions for later. I just want to do 2 final remarks. One of them is a bit of a different way of how to use models. So right now, we use models in a way to understand ecology, and we want to model ecology, but I want to say that we can also use models to understand methods, sampling methods. And I'm just gonna give you here an example, which is a one I know, probably there are more out there, but the MIT people, Stephanie Dudkiewicz and company, for example, incorporated a radiative transfer model into their plankton model, so into their global model. What this means is that they now simulate the trajectories of light and how particles absorb, scatter this light. And this allows us to simulate what is seen by satellites or by equipment that is used in the field that many times use optical proxies to make measurements. So here are 2 examples that I could find of how models are used to test or to understand what we measure. One of them is this, well, it's my own project, but down here, mainly what I did was to, since there is almost no phytoplankton carbon data, I said, okay, well, we have no phytoplankton carbon data and this data is biased. And also they have used very different methods to measure this phytoplankton. But we are using backscattering as a proxy for phytoplankton. And this is used in primary production models, satellite-derived primary production models are commonly used by many people in the field of ecology. So I thought, okay, can we really assume that backscattering is a good proxy for phytoplankton? We don't know. But what I did here was simply to calibrate an algorithm in the same way they derive it in real life. I calibrated to the model, to the phytoplankton carbon in the model. So this removes all the uncertainties of methods and biases, and then compare it and looked at the biases that you can get by using this algorithm. So here I can get a measure of, okay, backscattering is good or not good as a proxy for phytoplankton carbon. The other way here that is used was by B.B. Kale. It's still under review, I think, this paper, but what they did was so it has been said that you need at least 20 or 30 years to detect the climate change signal to detect whether there is a climate change signal or not in, for example, chlorophyll, satellite-derived chlorophyll data. So what they did is, okay, remote sensing reflectance, which is what is measured by satellites, basically, has less uncertainties than the chlorophyll product. So we're going to try to find out if we can get this signal from climate change in this product. So here, this is what they found. They said, okay, in these pink regions are the regions where the signal to noise ratio is high. And we have about, whatever, 20 years of satellite data. And then they said, okay, let's see if this is real, if this is a climate, well, if this is a climate change signal. And what they did was to take the remote sensing reflectance from the model, and they looked at the time of emergence. So how many years you need to be able to capture this signal. And even though you cannot see it very well, it's basically the kind of dark blue regions. So that's up to 20 years, and they kind of match the regions where they have like the pink. So it was a way to kind of test a bit the hypothesis. I mean, I'm not saying that it validates a hypothesis, but it gives an extra argument. And finally, the last remark I want to do is that, yeah, maybe it's also nice. We're all developing, well, all no, but many of us are developing our own nerdy models, and they are all very nice to use and all, but maybe we want to be able to share them with the field, or they need to be, they should be more widely used by the field. I mean, we cannot just keep them by ourselves. So right now, the NUM model we are trying to do this with the NUM model, especially this is led by Amalia Papapostolou and Ken Anderson at DTU, where they are making a user-friendly and fast-to-run model that you can run in your own computer without needing all these extra resources from the university and the cluster. I mean, if you use it, that's fine, it goes faster, but you can already run it in your own computer. So this is work that is being developed, and hopefully it will come out at some point. So keep an eye on the work of these people. And yeah, that's how I want to end this presentation. I have left here a few questions that I came up with for people to discuss if you want, or that we could discuss here. So I don't know, feel free to give your opinion, or if you have any question, or yeah, I mean, it's mostly to interact between fieldwork people, modelers, satellite, whatever that you think we need to improve. And that's it. Thank you very much.
Great. Thank you.